always, welcome to the 11th webinar. Yeah, today we're going to talk about scale, the big topic of scale in bioprocessing. Yeah, we're going to look at it from two different perspectives. Yeah, so Gerald Striedner will look at it more from a wet lab perspective, but also with modeling, and Christian Ritz will look at it more from the modeling perspective, but also, of course, with wet lab as the foundation. So I'm very excited about this. This webinar, as always, will be 20 minutes talk, followed by about 10 minutes Q&A. You can either raise your hand then in the Q&A or write a question in the Q&A field. Feel free to write questions during the talk. After 1 hour, we'll stop the recording. So with this, I will introduce our first speaker, Professor Gerald Striedner. He's from the Institute of Bioprocess Science and Engineering. He's also the head of this institute. This is at Boku University here in Vienna. He's also at ASIP, one of the area leads of the health biotechnology area, the other one being me. So we're very close colleagues, actually. And in the past, Gerald looked at scale issues in microbial fermentation a lot, and this is what he will present about today. So you're still muted, Gerald, but feel free to start your, actually I will put your presentation on screen, and I will put Christian and myself away. Perfect.
Okay.
Yeah.
Okay, so thank you for introduction, Nico, and welcome to everybody in the audience. As already said, I'm going to talk about bioprocess development and how this process is more or less involved also in these different scales. So whenever you do this process development, of course, you will use different scales. And when you have a closer look on that, what is happening, there is on the one hand side, the production and manufacturing scale, which is really big. Then there is lab scale, and then there are several options for very small scale experimentation. Whenever we have these small scale experiments, it's higher throughputs, it's lower costs. In most cases, you will have a bigger distance to the conditions that cells may see in the manufacturing scale. But of course, you can do much more experiments, and by that, this is super helpful. It's used not only in process development, but the small scale devices are already used for the work in host cell engineering. Whenever you don't have this idea also to have a closer, that later on you will have different conditions in production, you also have high risk that you develop hosts and select for hosts that do not perform well in bigger scale. And the distance, of course, between this very small scale to the production scale in terms of conditions is quite big compared to that what we have in lab scale, what we claim in lab scale fermentation, that we can directly go for scale up to production, even so there are still some effects that you cannot prevent. They are simply there. So for the agenda, I have decided to present two showcases. One is on a small scale cultivation system that we introduced into our work scheme to do screening experimentation. And the second one is on a 2-compartment scale-down bioreactor that has been used as a tool for process transfer from lab scale to manufacturing. So for the first topic, what was the motivation to introduce a small-scale cultivation system? First of all, the traditional one is the shaker flask, and it's quite clear that if you use a shaker flask, the conditions there are uncontrolled. They are quite different from that what cells see in a fed batch cultivation, for instance. This was one reason, and to get somehow access to higher throughput was driven by the fact that there was huge progress in molecular techniques and high throughput cloning. So these guys are able now to produce a really huge number of clones that have to be tested and characterized. On the other hand, there was also significant progress in the development of small-scale bioreactor systems and especially with high throughput and miniaturization. Whenever they are also more or less equipped with monitoring and control capabilities, they will get also more expensive. So it's a trade-off. You have to decide what you really need. And so we came to the conclusion that the bioreactor, which is a data plate format cultivation system, meets our requirements. So there is this defined conditions from temperature, humidity, and we also have a high shaking speed that allows us to also get high oxygen transfer, especially if we then combine this device with the flower plate, a special designed plate, which allows for even higher oxygen transfer. So we can get up to 10 gram cell dry mass per liter, which is quite high. Working volume is around 1 milliliter. There is also the option to do online pH and dissolved oxygen measurement via optodes, and there is also the option to you can always also follow the cell growth via scattered light measurement. And finally, what was most important to us is this feed media. This is a fed batch-like cultivation. What we can do there is that we can run the process like a fed batch with a carbon-limited condition. And this is done by using dextrin as the carbon source. And then there is an enzyme that has to be added and that releases glucose in a certain rate. And so this is kind of a linear feed profile what we end up with. So this is the starting point from the technical perspective, and most attractive or most important to us was then to verify if we can use this results, and for instance, if there is a screening result, if this is then also the same if we do experiments in bench scale. So is there transferability? Yeah. And for that purpose, we selected a concept where we have 4 different Fab molecules, relevant molecules for biopharma. And all these Fabs were designed with an identical amino acid sequence. The only variation was within the antigen binding region. And so only tiny differences in terms of the sequence. And we then did different combinations. So the 4 different Fabs into 2 hosts, E. coli strains, a B strain and HMS strain. Then there were 2 different signal peptides, post and co-translational, and genome integrated or plasmid-based expression. So in total, we end up with 32 clones. And we then did our initial experiments in the bioreactor, and the outcome was somehow a little bit surprising because there was huge impact from the individual molecule from the Fab side. Yeah. So FDN2 was the Fab, the black bars, what you can see here, always ending up with the highest titer. And FabX was the worst case scenario with more or less no product in the cell. And the behavior of the 2 strains was very, very similar, and the pattern was more or less the same. And what else was important is that HMS always showed some higher specific Fab titer compared to BL21, which is the strain that is most frequently used in manufacturing. So then we continued with our comparison, and usually the genome integrated clones perform better, sometimes 3 times more specific Fab in the cell compared to the plasmid-based one. So there is reduced stress because there's only 1 copy. It's a little bit higher stability that is given there. So we then decided to continue with the genome integrated clones and do the experiments in a benchtop scale system with these clones only. Yeah. And So it was standard fermentation process, a fed batch cultivation with an exponential feed, an exponential feed growth rate 0.2, so a little bit higher than that what we have in the small scale experiments. And the results were quite nice. So we end up with more or less the same ranking. Again, FDN2, the black bars, always the highest titer, higher concentration within the HMS strain, lower concentration of FAB in the B strains. And this was more or less a really nice confirmation of the transferability of the results, because you would came out with the same conclusion on the best performing strain. The main reason that there is higher growth rate in the dust skip fermentation was more or less the reason that there is a lower FAB titer. Yeah. So whenever you do these experiments, you can more or less use the device quite efficiently. Most important is that that the quality of results really strongly depend on the protocols, how to cultivate the cells, and also on the interpretation of results. And you need to know the cultivation systems characteristics, yeah, because for instance, there is to a certain degree there's impact on pH because of cell growth. pH in turn again impacts the activity of the enzyme. So there are some facts that you need to know if you want to use the system. And what else is there? Already tiny changes in the protocol can lead to significantly different results that can no longer be transferred to benchtop cultures. So you have to be aware, and we are, that you cannot always follow that concept, and sometimes you simply need to do experiments in larger scale to get a good idea of what is working and what is not. So this is first result, so first topic. The second one is now on this two-compartment scale-down bioreactor. The reason here why we entered this project was because we were in collaboration with the CMO, a contract manufacturing organization. So the company sometimes can do their own process development. And if it's that way, you usually have a quite good idea what will happen in a later stage if you do the transfer from lab scale to pilot and manufacturing scale. However, whenever you need to take over a process from another company, it's quite a lot of work to get this running. You stick to the protocols from there. Maybe there is no option to change anything. And sometimes it's also that way that this product is already on the market. And they simply move to the other company because of higher demand and need for bigger scale. And then there could also be impact on the product quality. Yeah. Whenever you compare lab scale and production scale, in our setting, it's that way that we have fed batch cultivation, which is the standard. You have addition of feed of acid and base and also of air. And by that, in a big scale, you will always produce a certain level of gradients, especially because of the media feed. As there is high glucose concentration, cells will take up the glucose, will consume more oxygen, and will also produce more acid. And by that, you will see these gradients built up in larger scale. And the second point is that there is longer mixing time because the efficiency in mixing is much better in a small scale bioreactor system. So the aim in our project was then to develop and construct a scale-down reactor, then to investigate scale effects on expression system and individual product, and then also to demonstrate that data generated in this scale-down system represent the outcome of a large-scale process. Yeah. And so there were certain considerations at the very beginning, because you can select for different scale down systems. For instance, you can simply do pulse-wise feeds, which is very simple, but not well suited for E. coli process. The same is if you install blades, it's also an option to interfere with the mixing efficiency in the lab scale system. However, the process that we have in use is that intensive that you cannot work with that. This is something that may work in cell culture processes, but not in microbial processes. So what we have had as design considerations then was first to go for a two-compartment system, a stir tank reactor connected to a black flow reactor. Then there was also, based on literature information, there should be about 20% of the total volume in the black flow. What we were interested in to have a system that can easily be sterilized, can be operated in a secure way. So especially how to connect these parts. And then we also installed options for inline measurement of pH, DO, or also conductivity in both compartments, the stir tank and the black flow. And there are also options for sampling from the black flow reactor and from the stir tank, of course. And all parts were sterilizable so that you can really work in good conditions. And for operation of the system, what we did is we get some information from the bioreactor system in the company, which is a 4 cubic meter bioreactor. And what they did already before is CFD simulations. And so what they have had in hand was already information on the mixing time. And we did the same then for the 30-liter system. I will not go into detail because the next presentation is much more focused on that part. So what we decided to select as a criteria is the residence time in the black flow reactor should equal the mixing time difference between the scales, which is quite a significant difference in the larger scale. At low stirrer speed, the mixing time was 49 seconds. In the small one, I think it was somehow in the range of 18 seconds. And during such a process, you will also see variation in the steering speed. So this may also contribute to the setting in the scale down system. And the adjustment of residence time can easily be done via control of the flow rate from the stir tank reactor to the black flow system. And the final outcome is shown here. So this is more or less on the left-hand side, the drawing and also a real picture of the system. It's very, very flexible. So it's made of stainless steel, so that the black flow reactor, there's quite a big pump that is used to do transfer of the cells from the stir tank and to the black flow and back. And then there are also sampling devices at the beginning, at the end of the black flow bioreactor system, and also measurement devices. For more detail, you can simply go for the publication shown at the here is from Maya Florian. It's on computational fluid dynamics. And we did a lot of characterization of both parts and also do measurements, how long it takes that there is, for instance, no oxygen left in the black flow, which is really that what happens if you then run real culture. Okay, so the questions to be answered in that project. So the initial question was, of course, if gradients in manufacturing scale impact cell growth, productivity, and are there differences in how so different production strains may respond to these effects? The second one was related to the product, because it's well known that norleucine can be misincorporated into recombinant target proteins if there is gradients and also oxygen limitations, which may take place in a big scale. And then, of course, most important to us, if we do these scale down experiments, is the outcome then representative for that? What will be the outcome in a large scale? For that, of course, you always need the company partner, either the clone from their side and the process, or they do the scale down experiment on their own side. Yeah. So for the experimental setup, again, Fab as the product, the best performing, of course, this FTN2 Fab, what I've shown before. It's genome integrated and into the two strains, HMS and BL21, because they are both quite frequently used in manufacturing. From the experiment, what we did is the scale down experiments in the 20-liter working volume stainless steel bioreactor, which is connected to the black flow. And for reference, we did experiments only in the stir tank reactor. The setting was high cell density fat batch cultivation with about 80 gram cells per liter at the end of the process. We started with a 9-hour exponential feed with a higher growth rate and then switched to a linear feed for 20 hours. And induction was done for about 12 hours in that process. Then we also did quite comprehensive offline analytics to get a better idea what is going on in these two different setups. So for the results, first, the cell growth. This is something that is quite well known, that whenever you have a larger scale, you will have gradients. Gradients always force more or less byproduct formation, and by that you will see lower cell densities. And this is the case for both strains. The blue line is for the reference from the stir tank, and the green one is for the scale down. So in summary, it's about 10% lower biomass in the scale down experimental setup. In relation to product, for the Fab production, what you can see, there was quite significant impact for the B strain, higher yield in the reference and lower yield in the scale down experiment. This is not the titer, but it's the specific Fab concentration per gram biomass. And not that much impact for the HMS strain. And what else you can see that in this case now HMS shows up with a little bit lower titer than I have shown you before. The reason for that is that HMS does not perform that well if you go for high cell density cultivation. So this is also something that is quite well known. So also a good explanation for the difference to that what we have seen in the former experiments in fat batch cultivations at lower cell density. What was more surprising to us is that in case of the reference, there was a higher rate of cell lysis at the end of the process. This and even higher in BL21 compared to HMS. So this is something that was really not expected. So indication that cells, even though they came up with a lower biomass yield, are somehow a little bit more robust. So they are well trained to continuously changing conditions. So this is at least one explanation for that. And whenever you see an increased Fab concentration in the supernatant, this is not because of passive release of the molecule to the supernatant. This is usually always connected to lysis. And this will also be confirmed in the next slides a little bit later. So for the misincorporation of this non-canonical amino acid, noleucine, this is an amino acid that can be formed in cells whenever there is limitation with oxygen and high glucose concentration, as already told before. So for that, we established a protocol to first extract the Fab from the cells with EDTA. Then we did an affinity chromatography for purification. Then there was hydrolysis step, and finally HPLC for amino acid quantification. And the results were a little bit also somehow surprising because there was more or less no misincorporation in the B strain. So this is red and orange line. in both settings, scale down and reference, but closer look shows that the HMS strain, there was significant misincorporation of norlucin, about 20 up to 15-20% of all possible positions where norlucin can be introduced into the protein. So this is quite a severe impact. And having this in hand, if you do the development at small scale, or if there is a definition how the product should look like, and if you then do a scale up to much bigger scale, you can run into the problem that the product maybe don't meet the criteria any longer that you have defined before. But finally, whenever you have the idea to go for production, in this case, BL21 would be the strain that you should use, because this strain don't show any problems with this misincorporation. So the next question now, what is about viscosity and DNA in the supernatant? So we did viscosity measurement of the suspension of the supernatant, then also the DNA measurement. And what comes along with that is whenever there is more DNA in the supernatant, and this is valid for the reference in P strain, this is strong indication for cell lysis. And whenever you have cell lysis and DNA, there is also an increase in the viscosity. On the other hand, there was one result that was not explained by that, and this is for HMS with the viscosity in the cell broth. This was not expected and cannot be explained by increase in cell lysis. The only idea that came up was that this is somehow related to a change in morphology, yeah, because otherwise there is no other impact factor that could somehow increase the viscosity of our suspension. So we then did additional measurements via the lumicizer, and what you can see here is that there is a shift in the settling velocity if we do scale down experiments, and a shift to higher settling velocity. We then continued with our study in that direction, and we did also scanning electron microscopy for both strains in both settings. And what you can see here in this microscopic picture is that the scale down experiments, the cells look a little bit more smooth compared to the cells in the reference system. Yeah, this is somehow maybe also related to that what we have already seen before, that there is a higher tendency to cell lysis in the reference system. We then also did dynamic light scattering measurements to more or less get an idea on the hydrodynamic radius of our cells. And whenever there is scale down experiment, the dynamic radius is much higher compared to that what we have in the reference experiment. So there is quite big change in morphology. And if we combine all these measurements, so we did the SEM and the lumicizer and the DLS measurements. The SEM is the gray root that is shown here, simply the length, the diameter, and also the area of this particle, surface area. Then there is from the lumicizer the blue circle. There's big change in size if you compare scale down and reference, and for the DLS measurement, it's more or less the same thing. So there is quite big impact on cell morphology, which was also not known beforehand, and this is something that we really see as a very attractive and interesting result from this study. So to summarize now, if you do this scale down, if you run scale down experiments, what you will see is lower biomass yield. You will have even higher Fab fraction intracellular. You will have reduced cell lysis compared to the conventional stir tank. What we saw is individual response of host strains to scale down conditions in general. And in context with Norlucin, there is B strain that shows up with better features here. There is no misincorporation in this strain. We also saw altered viscosity, higher settling velocity, and changes in cell morphology. And most important to us, and I'm unfortunately not able to show the results, is if results can be transferred back to that what is the outcome of a large-scale cultivation. These experiments were done at the company, and it took them some time to get familiar with how to operate the scale-down system, and it was not 3 experiments, but only 2 with the strain, where they also have had the information on the large-scale results. And what they saw there is that the small scale or the scale-down system really is able to came up with really the results that you will see later on in the big scale. And from that, of course, there's huge potential to save a lot of money because you can rely on data generated in a very small experimentation, and you don't need to do too many experiments in large scale to show and demonstrate that you're able to run the process accordingly. So this is from my side. And of course, I'm now open to take your questions.
Great. Thank you, Gerald, for this really interesting presentation. So please, if you have any questions for Gerald, write in the Q&A or raise your hand so we can call you up. In the meantime, so you presented now on microbial fermentation scale effects, right? So what are the main differences compared to mammalian cell culture? So between microbial fermentation and mammalian cell culture, is it just oxygen uptake and
No.
substrate consumption? Or?
The biggest difference is the way how you run the culture. In microbial systems, you usually go for carbon limitation. So there is 0 glucose in the process, and whenever you add glucose via the feed, then you will generate a quite significant gradient. In the cell culture process, you are not allowed to go to 0 with the carbon source and also with amino acids. So by that, it will not have that big effect, even so there is a feed. Yeah, because from a physiological state, the cells don't react to. Whenever there is carbon available, they will not be that reactive on that. Yeah. The mixing time was our scale criteria. This is maybe also not that what I would go for a bigger scale in cell culture, because I think it doesn't make that much sense. And also the setup of the scale down system, it's maybe also different, because whenever you use a pump in such a setting, it's maybe not that beneficial for mammalian cells. Yeah. But what
this was a peristaltic pump, right?
Yeah. What I know, and it was running at quite high speed, so it was really loud during the experiment. Yeah.
And actually, a more technical question. So, what was the volume of the plug flow reactor relative to the actual reactor?
About 20%.
Okay.
So this is something that is more or less stated in literature, that you should have this distribution more or less. Yeah, that this is something that is representative. I don't have any idea if this is now correct or not. Yeah, but this is what is in general accepted by the community.
Okay. So any questions from the audience? I see we have a very shy audience, just like last week. This is very strange. Yeah, in the past the audience was there's 30 people here. I'm surprised that no one has a question. So this viscosity difference, you think? So because you showed one experiment that had differences in cell lysis right? Where the scale, the larger scale or the scaled up model showed lower rates of cell lysis, but then it also showed higher viscosity.
Yeah, but only for one strain. Yeah.
Okay.
But this was for HMS because there was quite significant change in morphology. Yeah. Usually, whenever you have this cell lysis, of course, DNA is released and you can expect higher viscosity. This is clear. Yeah. What was not that clear to us is that in this scale down setup where cells are exposed to higher stress level, that these cells are somehow more healthy compared to the other ones. Yeah. Because the degree of lysis was lower, significantly lower, or at least no lysis.
Okay, then there is a question. I don't fully understand it. How to mimic glucose-limited fat patch in MAP, but it's spelled MAP, or maybe it's MAP, like monoclonal
What is
antibody.
MAP?
Yeah, I'm not sure what is MAP. So maybe it's a monoclonal antibody is spelled
But maybe
CHO cell culture. But there you don't have glucose-limited fat patch, no?
so. Maybe the question
It's
is
a different
related to
map.
the small scale. I'm not sure on that. But of course, carbon limitation, what we generate in this small scale environment, all the dextrin is already added at the very beginning. But then there is a certain amount of enzyme that is added, and by that you can more or less get control on the glucose that is released per time. And whenever cell density is high enough, they will consume, and then you will also get somehow control on the process in that way.
Okay,
but
yeah,
I don't really know what is meant by
Susanne
MAP.
clarified. Oh, so it was a typo, MTP, microtiter plate. Yeah. So
Yeah,
basically, yeah, the enzyme, the starting substrate, I guess, of the dextrin and the enzyme concentration dictates the feeding rate then
yeah.
or the
For the
release rate
batch,
of the glucose.
for the batch phase, there is a certain amount of glucose that is provided. It must not be too much because otherwise you will run into problems with oxygen limitation. Yeah, if cells can grow
Even
in
in the
batch
small
phase.
plate.
Yeah, even because when cells can grow to higher cell density in with a high growth rate, you will see limitation. So you have to be careful in that sense, not to add too much glucose to the starting batch and then simply stick to the release from the dextrin.
Okay, I hope that answers Susanne's question. With that, we're over time already. So
Yeah.
with that, I will pull our next speaker up to the stage. Yeah. So yeah, thank you, Gerald, for your talk. Now, Christian Witz is the CEO of Simvantage. Yeah, he did his PhD at the Technical University Graz in basically also related to computational fluid dynamics, and then spun out his company Simvantage there. And he will tell us now about the modeling aspects of scale aspects. With that, the stage is yours.
Thank you, Nico, for this nice introduction. Yeah, here are the slides. I hope you can see them, and they changed the picture now. So I will jump right into the topic as we are already at what we heard over time. So what can simulation add to bioreactor scale-up? We have already seen how scale-up is done in a very sophisticated manner. But the first thing that a simulation can add is to obtain a complete picture from individual sensors. As Gerald has shown, that mixing time is often used as a scale-up criteria, but it's measured with one sensor sticking into the reactor on the side. And if you're lucky, then these sensor measurements represent the actual mixing time. If you stick it in a zone where there is very fast mixing and where it's very quick to reach the desired homogeneity, then you get a false image about the actual mixing time. And if you then compare reactors like a small scale and a large scale reactor, this effect can even get more pronounced and can lead to the wrong scale up parameters. So from the simulation, you get a full picture of the reactor, and you can use the sensor value for validation, but you use all the volume for determining the mixing time. The second thing that simulation can add to bioreactor scale-up is that you get insights into otherwise inaccessible parameters like the shear rate or the energy dissipation, which is very important if you think about mammalian cells or cell agglomerates in case of the energy dissipation rate. And with simulations, you all get all these parameters easily out of the resulting data. And of course, what you can also do is reduce your experimental effort because you save a lot of time. The setup of these virtual experiments is very fast compared to building a whole reactor in the lab. And you save money because you always use like media, person hours, and it's much cheaper to do it on the computer with graphic cards that are available and that are relatively cheap in comparison. You can also think about sustainability. This means the material saved from the real experiment is also adding to your sustainability balance. So let me give you a quick overview about what can be done for a bioreactor. So what simulations results can you expect, and what can they be used for? So this is, of course, a slide about our software, but it's very comparable for other software packages on the market as well. So the first thing you get is the fluid flow field. So how the fluid moves inside the reactor, you get all the velocities, the shear stresses, which are important as we just heard, and also the steering power. Steering power is important for very large reactors. And because you want to save energy for steering in order to bring the operational costs down. Then for the gas phase, we can determine the holdup, which means what fraction of gas is in the reactor, the bubble size distribution, and with the different bubble sizes, of course, you have to take into account coalescence and breakup. And with these different bubble sizes, and you can also determine the KLA value, so the speed, how fast oxygen transfers into the liquid. And it can also determine the flooding point. So if you increase gassing more and more, at one point the stirrer cannot radially distribute the bubbles anymore. This is the flooding point. The bubbles then just go as a plume to the next stirrer or to the reactor top, and then you lose a lot of potentially dissolved oxygen. And then we can also include virtual cells, and these virtual cells then tell us what they feel when they float through the reactor. These are called lifelines, and based on these lifelines, you can do a very scientific first principle based scale-up, because when the cells experience the same microclimate, the same lifelines, in your lab scale reactor and in your manufacturing reactor, then it's very likely that you get the same results in both cases. You can also add particles that can settle, where you can determine suspensions, the just suspended speed, and also dissolution, like in this case, you see calcium chloride particles that dissolve slowly into the liquid. And if you think about microcarrier processes, you can also determine with your virtual cells the impact rate of these virtual cells onto these microcarrier particles, and then determine how to get the best initial growth phase on these microcarriers. Now, if we see this calcium chloride, which is like distributed to the liquid, we need to transport it around. Of course, it doesn't stay in place. And with this transport algorithm, you can determine mixing times, dead zones in your reactor if you have a certain consumption. And you can also do like oil droplet distributions. If you're using oil droplets as nutrient, you get the droplet size distribution and also how they are spatially arranged. One important thing, of course, is the temperature inside the reactor, and you always run into the problem that you don't know how much energy goes from your heating jacket or cooling jacket into the reactor volume, because you need to determine like the energy balance between the inlet and the outlet of the heating or cooling media, and this is usually not available in the plants. And therefore, we developed, as you can see here, a two-fluid simulation. So you simulate the reactor, the inner reactor, where you have your fermentation broth or your cell culture media, and you also have the heating or cooling liquid, and therefore you only need the inlet temperature and the flow rate. The rest is calculated. And now you may ask yourself, okay, you've now shown us fancy pictures, fancy videos. Is this done with this new AI thing, or is this the real physical reality? And for that, you always have to look at validations. And if you take one thing from my presentation today, then every time from now on, when someone shows you fancy pictures from simulations, ask for validations where you compare experimental results to the simulation results in the same system, and therefore you know if the simulations that were done in a manufacturing scale, for example, are valid. And how this can look like, I would like to demonstrate here. So you have here this tank from the publication of Xi et al. For 30 liters, 100 to 300 RPM range and a certain gassing range. And you compare the mixing times. And this is a typical graph where you have the experimental values on the x-axis, the simulated one on the y-axis. And if you have like 30 seconds simulated and 30 seconds experimental, so a perfect match, then the point should be on this 45-degree line. This would be a perfect match between simulated and experimental value. The farther away the points are from this 45-degree line, the more error is in the simulation. If this error grows too large, then you have to question the engineering office or the guy who just showed you the simulations, whether this simulation technology can be trusted. But here you see a diagram with really good results and very well validated. This can not only be done in the small scale, it was also for us important to have like a data point in the large scale. And this validation was done in a 160 cubic meter reactor from Sandoz Novartis Kundl in Tyrol. This was the time when Sandoz and Novartis were still together. We had this reactor available. It was a reactor with 4 Russian turbines. We had 2 probes, 2 conductivity probes, a lower and upper probe. And then a salt solution was injected from the top and then slowly homogenized throughout the reactor, slowly was distributed. We did a digital copy of this reactor in our software, injected a substance from the top and placed 2 virtual probes inside the reactor. And then we compared the measurement signal from the CFD simulation and the measurements, the 2 measurement runs which were taken. And you see that for the upper probe and for the lower probe, you have a very good agreement between measurement and CFD simulation. This was not the end of this project. And then the engineers from Novartis started playing around with the steering system on the computer. And they tried out different configuration, different steerers. And they found that changing out every second steerer stage to a pitch blade configuration resulted in a reduction of energy demand and a faster mixing, so a reduction of mixing time. They changed out this configuration also in the real reactor. And they saw they could really reduce the energy demand by 20%. Saving this, now it's a bit more because of the energy costs, about around $500,000 per reactor per year. They have 10 reactors there. They changed it out in every reactor now. So they have quite significant savings in operational costs while having a faster mixing in the end. And this was also published in BNB 2022. And the question you may now have is that I'm showing you, I'm using my precious time here to show you validations. Why am I doing that? First thing, I told you before, it's always important that you are sure that simulations show you a correct result. And the second one is that you need these validation cases to integrate this simulation data also into regulatory submissions. And the good thing now here, where I present you this first principle CFD solution, is that if you're doing first principles of basic physics, you can do a validation at one point in one reactor. And if you have another reactor which is a bit bigger, a bit smaller, has a different operating conditions, these validations are still valid, and you can still use it for doing the regulatory submissions for this reactor, which is different to the one where I did the validations in. Because the physics does not change if you change the reactor from a cylindrical reactor to a cube reactor, or if you change a Russian turbine to a pitch blade turbine. The physics does not change, and therefore your validations are still usable also for your use case. And if you're interested in how especially FDA assesses these simulation data, I can point you to this guidance. Of course, it's the credibility of computational modeling and simulation in medical device submissions, but the credibility assessment that is described in this guidance is also used internally at the FDA for manufacturing. And this credibility assessment is taken from the SME V&V40 standard, and there is currently a working group, which I'm part of, to develop the V&V80 standard, which is then specialized on manufacturing and has certain use cases included so that you know how to do this credibility assessment also for manufacturing devices. Now I would like to take you a bit on our journey, how we developed our scale-up software. And the first step of that was actually a governmentally funded project, also done together with the BOKU. And it was the program Project eConti for the continuous bioproduction with E. coli. And let me show you a quick overview. In the whole continuous production process was for the production of protein A as model protein. The cells were provided by engines. They modified it in order to have two reactors in this production chain. The first one is a growth reactor where you don't produce any protein A, but you produce biomass. Then the production of biomass is stopped in the production reactor, and the production of protein A is switched on. Therefore, you avoid the problem of having overgrowing cells that are not producing. They are overgrowing the producing cells. And then all this equipment was put together and connected with the software Cubicon. And this software was then on the digital side aided by Novasign, who did the simulation for the biological part, and our part, which was for the physical simulation. And as Nico is already here, I assume I don't have much time left. Is that correct?
Well, we have 5 minutes left in the meeting, then the hour is over.
Okay.
I am not entirely certain what will happen after the 5 minutes are over.
So, okay, then I will cut it short. You don't see the fancy video we have produced about this project, but imagine this whole setup in the...
We did have a presentation by Wolf and Jürgen from Cubicon and from Engines. So
great.
We did cover this, I think, in February.
That's good.
But of course, from a different perspective.
The point I want to make in this video is that you see here this window, which is kind of important, where you see the process parameters like flow rate and steering speed on the left side, and the mixing time, hold up, and average shear rate, which comes from our model on the other side. And this is done in real time. So you might ask yourself, how can a simulation, which takes usually a few hours, be so fast that you can have real-time results? And I would like to show you how we did this. First, we took the reactor system, and these are the operating ranges, like for the broth weight, steering speed, and gas flow rate. So you have this three-dimensional space, and in this three-dimensional space, you now do a lot of randomly chosen parameters. You do CFD simulations. So for each of these random dots you place in this three-dimensional volume, you generate out of the steering speed, gas flow rate, broth weight, can also be viscosity, and so on. In this case, shear rate, KLA, and mixing time. And then you have a lot of these reactor data, but they are individual dots. And then you use a so-called rapid response model, machine learning algorithms, to interpolate between these dots so you get the full picture. So now for this reactor system, for every operating condition, we throw at the rapid response model, it gives us within a few milliseconds the shear rate distribution, the KLA, and the mixing time. And with this rapid response model, we now can do scale up. And this is done in the following way. We have our source reactor, like a lab reactor, and we are using our rapid response model to get from the operating conditions in the lab the physical parameters. I always choose these three parameters, but of course there can be more, like the shear distribution, the mixing time, and the KLA value. And now we go to the target reactor, where we also use our rapid response model now for the target reactor, but we use it the other way around. So we take an optimizer that randomly guesses an operating condition and then compares the result of the shear rate distribution mixing time KLA with the one we would like to see, which comes from our source reactor. And then it does a gradient descent until it reaches a point where you have the same physical parameters in both reactors, and then it gives you the operating conditions for the target reactor. And to make this more tangible, here a proof of concept where you transfer operation from 2 liter to 100 liter. So in the 2 liter, you say you have 500 RPM, a certain gas flow rate, and a certain broth weight. With the rapid response model, you end up with this KLA, the mixing time, and the shear rate distribution. And now you want to know for the same KLA, the same mixing time, and the same shear rate distribution, what do you need for the steering speed and gas flow rate for this 100 kilogram of broth weight. And then the optimizer just guesses 107 RPM and 1.6 cubic meter per hour. And you see there is quite a distance between the reference distribution and the target distribution, also for KLA and mixing time. And then what you can do is, after 20-second iterations, he has found, the optimizer has found a perfect match. And it gives you the operating conditions for 336 RPM and 2.2 cubic meter per hour. Then you get the same KLA and the same mixing time. And that this works was recently published together with the RWTH Aachen in this paper, where Streptomyces species was scaled up with the P95 energy dissipation rate, and they saw that the morphology, the cell morphology between the cells were much more comparable if you do it with this method compared to the traditional power per volume scale up. And the last slide I would like to show you is that we packaged all this together into a cloud-based solution where you can set like your lab reactor, like the Amber 250, and the target reactor, like a STR50. And then you set what you did in the lab in terms of steering speed and gas flow rate. Then you set the ranges for the target reactor where you want to operate it in. And then you select your scaling criteria, like the KLA, a P90 shear rate distribution. And then you click Run Scale. And then you immediately get the operating conditions for the target and also a comparison of the scaling criteria. And you can also compare like the shear rate distribution, the Kolmogorov length scale distribution, and so on for both of these reactors. And the last thing you get is you can visually compare the two reactors with each other at the point that the scaler has recommended to you. And this is the last slide. With that, it's a bit of a tour de force. I would like to give you an outlook. So we can also simulate shake flasks now and well plates. So you can start your in silico scale up with shake flasks. You can simulate the light intensity also. So you can also do photobioreactor scale up. And we want to extend that to filtration, lyophilization, freezing, all the unit operations that are there. With that, I'm at the end, and I hope for a lot of questions. Thank you.
Yeah, thank you, Christian. Yeah, really interesting talk. Also, always nice to see. I really like seeing the simulations. They're always very visually pleasing. So please, I hope there will be a lot of questions. Raise your hand or write in the Q&A. In the meantime, so you showed this example from the 160,000 liter reactor, right? So do they use a scale down model there? And I'm curious, what scale is the scale down model of a 160 cubic meter reactor?
So they have a pilot plant facility. I'm not sure if I can disclose anything about
okay.
this. Yeah,
it's too bad.
But in this project, also this wasn't the target. The target was that they had an established process which they wanted to optimize in terms of energy efficiency. And they also posted it their success on the Earth Day on LinkedIn. That they now are so sustainable because they reduce the steering demand. I mean, they have a 0.5 megawatt of steer on this. It's quite a lot of energy, of electrical energy. But unfortunately, they didn't include our name in this post.
Okay.
Yeah, that was a pity.
So, you showed 20% energy savings just by changing the impeller shape, right? So from Rushton to the other one. So what is there a rational explanation to this, or was this only accessible through simulation? Like I'm actually quite surprised. That's a huge difference, right? And seems like a small change. So what's your explanation?
For a Rushton, if you imagine a Rushton, so you have this disc and these plates.
Yeah.
And you have these two circulational vortices. And the transfer between these two vortices is very restricted. So at each disc of the steerer, you get like a stopping zone for the mixing.
Okay.
And if you now use this pitch plate, which is like up or down pumping, you kind of destroy also this this boundary
in between.
Yeah, and not only in between, but also for the upper and the lower Rushton that you keep, because it's always advisable to keep at the lowest position a Rushton turbine, because this sets your bubble size distribution. And but for the upper ones, it's always advisable to play around with radial and axial steerers, and not only keep one type, so to say.
Okay. So please, from the audience, a plea to the audience. Yeah, please post some questions, or maybe Gerhard, if you have a question. But actually,
yes, I have one.
Perfect.
And the question is related to the last example that you showed. Yeah, that you can more or less get quite nice information how to operate the larger scale bioreactor to get the same mixing time, to get the same shear force and KLA. But I expect that there's a certain limitation in the scale that you can go for. If the first one is maybe in 1 liter, what would be the upper limit? Because from 1 liter to a 10 cubic meter, I would expect you cannot get the same settings. Yeah, you will not reach that.
This is actually also reflected in the user interface. I was not able to show it in detail, but what you get is also this percentage range on the right side. where you have the comparison between, okay, these operating conditions in the small scale, this operating conditions in the large scale, and then it shows you a percentage how well these fit together. And you get also like it shows you what the mixing times and the KLA are. And if you like choose a very large reactor, of course this percentage will go down, and it will tell you, okay, that it writes it down also. It will tell you, okay, this mixing time you cannot reach with this huge step. You cannot reach a good agreement between the mixing time, for example, because of course, if you go from an amber to a 160 cubic meter, the mixing time will always be different no matter what you do. Yeah, and yeah, basically that's how we reflect this problem with a like a visual thing that you see how well this fits together.
So are there specific scaling criteria that usually go well together and some that don't? Because I mean, you can use different scaling criteria, right? Like KLA or the mixing time or whatever. Like, and when do they fit together and when do they actually not? I assume that they don't. It's not always straightforward, right?
So what we have seen is that it's not like this geometrical similarity, but rather you have to have a certain stereo configuration, for example, which is kind of comparable. For example, if you think about like the amber with its stereos that are mounted in the middle of the reactor, two stereos, which sweep a really a large volume compared and they are distributed. And then think about the XTR 2000, I think, which has like a bottom-mounted stereo. And the thing is, oftentimes you only notice it when you have like non-Newtonian viscosity, because then the amber with its two stereos can produce enough shear rate to get the viscosity down in the whole reactor, whereas the XTR 2000 has a hard time to get enough shear rate to the top in order to have a lower viscosity on the top. So
okay, that's fascinating. Yeah, we do have a question for Barbara Petschacher. Thanks a lot for the very interesting presentation. In general, what are the greatest challenges you meet when running such a project, like scaling up from small scale to production scale in real life?
Yeah, I mean, so it depends on the perspective. The greatest challenge for us while doing such a project is to get the right geometries, because oftentimes they are like legacy reactors, which are there for an infinite number of years. Nobody knows really how what their current status is. We had people that needed to go in the plant, look in this small window in the reactor and look what stereo is actually in the reactor.
Because no one knew.
Yeah, because they had like the drawing was in conflict with what you saw in the reactor. And so this is always a challenge.
This is an unexpected challenge, yeah.
Yeah, we always tell this that this will be like the most time-consuming part of the project, and everyone is like, nah, we have that. Yeah, until they need to send it, and then this is something. The other thing is that sometimes you really reach the conclusion that it's not advisable to do this scale up. So we had one CDMO where we helped with this scale up. Do you say train? Scale up train? I don't know. And they used this very specific reactor in the middle. And we said, okay, the smallest one and the biggest one you have is really comparable, but the one in the middle is not. And they said, okay, we will take care of that. We will ask why it is used. And then after 2 weeks, when we had the next meeting, they said they asked it around. Nobody knows why this specific reactor is used. And then we actually proposed a more fitting steering system for their middle reactor in order to have a more comparable and stringent scale up.
Probably just had a reactor sitting around and used it.
Yeah, I don't know. But these are like the challenges. Of course, we are not so much in the biological space. So of course you always have challenges, and we often do this together with the company Novasign I just showed, because we can automatically exchange data between their hybrid models for the biology and our physical models for the reactor. And this is really fruitful for the scale up, because you see both sides, so the biological side and the physical side and the CFD side.
Yeah, I mean, yeah, the model you showed earlier, I mean, that's also a hybrid model then, right?
From us, it's not the hybrid model. It's from
no,
Novasign.
But this, you had a slide earlier with the A and N. There was an A and N part of a model, right? Because at that point, the model is fast to compute, right? Because then it becomes a hybrid model, no? This one, yeah.
No.
The rapid response model, no?
No, you wouldn't call it a hybrid model, because what you do, the CFD simulations on these points, let's say at 100 RPM and 110 RPM, and you get for both the shear rate distribution, and then you want to know 105 RPM, and you have not simulated it. And what this machine learning algorithm does is to do a multidimensional interpolation to this point, so that you know when you're setting up 105 RPM, also know the shear rate distribution. And that's a multidimensional problem. Therefore, it's a big problem than just linearly interpolate between those 2 points where you have the simulations.
But it's just interpolation based on a completely mechanistic model. Okay.
Exactly.
Yeah.
Exactly.
Okay. Great. Yeah. I mean, now it's almost 5, so maybe since there are no further questions.
There's one.
There is one?
Oh.
No, I think this is the one we yeah, there is one now. Okay. Yeah, could you with the system also see how to run the small scale reactors to resemble the large scale best? So in order not to overestimate possible outcomes. How to run small. Yeah.
So what you probably mean, please correct me, is we thought about that you do it the other way around, that you say, okay, I have this large-scale system at a specific operating point or a specific operating range. And now how does this translate to an operating range in my small-scale setup? So you know where your Amber system, for example, needs to be operated in order that you can then, in future, translate it to your large-scale system. So that you don't the Amber can spin very fast, and if you use a very high steering rate, this can never be translated into a large-scale system, and therefore you can. We are planning to do a tool where you do it the other way around, that you say, okay, I have this range in my large scale. What does this translate to in my small scale?
I hope that answers the question, Barbara. Okay. Then let's come to an end. Yeah, I want to thank both of you for the great talks, and thank you all for joining. And yeah, we will post about the next webinar. We don't have anything planned yet. So thank you, and have a nice day.
Thank you.